So before Neil introduces our distinguished speaker, I just want to personally welcome you, Eric, to the department. As Neil will tell you, we are having the pleasure of a visit from really one of the most distinguished and consequential MD, PhD psychiatrists, neuroscientists, not only in the country, but I think in the universe. So I don't think I overstate that. And we're really pleased that you have carved out a little time in your schedule to see not only us, where you have, I know, colleagues and friends, but also the broader neuroscience community here. So a warm welcome. Thank you for being here.
Thanks. Welcome, everybody, this morning. The silver lining for this storm is that it brought us Dr. Nestler. Eric is the Nash Family Professor of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York, where he's Dean for Academic and Scientific Affairs and Director of the Friedman Brain Institute. He completed most of his training and early career at Yale, receiving his BA summa cum laude, his PhD under Paul Greengard, his MD degree, psychiatry residency and fellowship, and board certification in 1988. He served on the Yale faculty from 87 to 2000, finally leaving New Haven in 2000 to be Distinguished Professor and Chair of Psychiatry at UT Southwestern, until returning to the civilization of the I-95 corridor in 2008. The goal of Dr. Nestler's research is to better understand the molecular mechanisms of addiction and depression based on work in animal models, and to use this information to develop improved treatments of these disorders. He currently is PI of a P50, a P01, 3 R01s to pursue these studies. The CV lists 380 plus peer-reviewed papers, 200 plus chapters and reviews, 7 books. He serves on numerous editorial boards, been deputy editor of biological psychiatry since 1997, also serves on many important advisory boards in our field, a member of the NARSAD Scientific Council since '97. His contributions have been recognized with virtually all of the major prizes in our field. I'll just name a few: the Oneida Merit Award, the Goldman, Rakesh, and Falcone prizes from NARSAD, the Efron and Julius Axelrod prizes from ACNP, the Sarnat Award from the Institute of Medicine, the Gold Medal Award from SOBP, and I'd keep you here all morning if I kept going. He's a member of the Institute of Medicine, a fellow of the AAAS, past president of ACNP, and current president of the Society for Neurosciences. So today he'll speak to us about transcriptional and epigenetic mechanisms of depression. Please join me in a warm welcome for our grand round speaker, Dr. Eric Nestler.
Thank you. Thanks. Thanks so much, Neil. It's a great pleasure to be here in San Diego. In fact, it was really fun to see the name given to this seminar room, the Marilyn Farquhar Room. Marilyn taught me cell biology when I was at Yale in medical school, so it's really a warm welcome in particular. What I'd like to do today, different from my title, is to give a broader overview of some of the work we've done in studying transcriptional epigenetic mechanisms of depression. And then I'll do a deeper dive into sex differences in depression, which was the original title of my talk, in my neuroscience seminar this afternoon. So a shameless advertisement for my second talk here in San Diego. So let me just start by putting the work that we do in some clinical context. I know as a psychiatry department, you all know this far better than I do. It's impossible to overstate the impact that depression has on humanity. It's a prevalent disorder. It's one of the world's major causes of disease burden and disability. Yet despite that, and despite much study in the field, we still know relatively little about what causes depression. It's been estimated that perhaps about 40% of the risk for depression is genetic, but there are no causative genes known. And just now we're beginning to see weakly contributing risk genes being identified in large GWAS studies encompassing hundreds of thousands of individuals. So understanding that genetic basis is going to be difficult and highly complicated. We know that stress is implicated in depression in some people, but less so in others. And we figure that depression now is a heterogeneous disorder, a compilation of many different disease states and likely different pathophysiologies. Now, despite the fact that we have learned relatively little about what caused depression, we do have some pretty good treatments. On the other hand, all of today's antidepressants are based on serendipitous discoveries now 6 decades ago. All of today's antidepressants have the same mechanism of action pretty much as those older medications. And we know from the STAR-D study and others that only roughly half of all people with depression are fully treated with today's medications and psychotherapy. So there's a very large unmet need that has defied investigation over several decades. As you all know, we diagnose psychiatric syndromes solely based on behavioral abnormalities, so I think it's all good for us as psychiatrists to take stock of that in 2017 we still lack an objective genetic test, blood test, brain scan that can be used to diagnose a psychiatric syndrome such as depression. It's sobering. This is an abstract of some of the symptoms that go to a diagnosis of major depression in DSM, and I just want to highlight one particular point. When we as psychiatrists make the same official diagnosis of major depression, whether a person eats too little, eats too much, loses weight, gains weight, sleeps too little, sleeps too much, has low energy, or too much energy, we know we have a problem with diagnosis, right? And it underscores the point that depression is a heterogeneous syndrome and needs to be examined in that perspective. We've learned a lot over the years in terms of the brain, areas of the brain that are involved in regulating mood under normal conditions and presumably where the pathology for depression exists, largely confirming 100-year-old lesion studies identifying so-called limbic circuits in the brain being important in these phenomena. That's shown here in a cartoon of a sagittal section of a human brain, including broad regions of prefrontal cortex, an area of the brain that I'll be focusing on a lot this morning called the nucleus accumbens, in the temporal lobe, the amygdala, and hippocampus, and many other brain regions, including the hypothalamus and the brain's monoamine centers, which are presumably the reason why today's antidepressants, all of which have a monoamine basis, produce their clinical effects. And so by analogy with every other branch of medicine where advances are made by the study of animal and cell models, how do I, as a molecular psychiatrist, try to study molecular causes of depression? And I think that this really highlights one of the cardinal limitations that we have in psychiatry. Since we can only diagnose a psychiatric syndrome based on behavioral abnormalities, and those behavioral abnormalities, many or most of them, are inherently inaccessible in a laboratory animal, it makes it extremely difficult to identify bona fide mechanisms of psychiatric symptoms. Nevertheless, I would, if that were it, I would stop now, but I do think that judicious use of animal models can teach us something about psychiatric syndromes, and I'll tell you today, give you an overview of how we've approached that challenge. So one of the models that I'll be focusing a lot on this morning is called social defeat. It was introduced into the lab by Olivier Breton, who's now at NIDA, and Nadia Tankova, who's now at Mount Sinai. And what they did was the following. They took a genetically inbred C57 mouse. They placed it in the cage of a bigger, meaner mouse, a retired CD1 breeder. Fighting ensues instantaneously. It's ferocious fighting. We limit it to just a couple of minutes to limit physical injury, and then house the mice for the rest of the day separated by a screen. So that test mouse is still subjected to all the aggressive cues of the larger mouse. And then we repeat that process every day, typically for 10 days. And what we found is at the end of the 10 days, we have induced a behavioral syndrome in these animals. And the behavioral syndrome is characterized by a list of symptoms. This would sort of be the equivalent of mouse DSM social defeat, right? And so we induce a very profound degree of anhedonia. I actually think we can measure anhedonia pretty well in a mouse. The mice don't show as much interest in drinking sugar water, eating high-fat food, having sex, running on an exercise wheel, and so on. We interpret that the mice have anxiety-like symptoms, but again, I don't know how I'd ever know if a mouse is anxious or not. All these measures rely on simply seeing a decrease in exploratory behavior. There is a hyperactivity of the hypothalamic pituitary adrenal axis, disrupted circadian rhythms, increased addiction liability, metabolic syndrome. That's really interesting because these mice, even though they don't seem to enjoy eating as much, if given free access to food, they will overeat and induce obesity in themselves and actually induce a pre-diabetic state of central insulin and leptin resistance. and then a profound social avoidance. And I'll be focusing mainly on those social avoidance data as a measure of social defeat in my talk. Now, we're particularly interested in social defeat, even though it's one of several chronic stress paradigms that we use and think are important, for the following reasons. Only about 2/3 of C57 mice develop the syndrome that I showed in the previous slide. The remaining, and we call them susceptible. The remaining 1/3 develop equivalent decreases in exploratory behavior and some other measures, the HPA axis measures. However, they do not show the range of other symptoms, and we call them resilient. They're able to maintain normal function in several behavioral domains despite being subjected to the same degree of social stress. Unlike most rodent models, certainly mouse models of stress where the animals recover very quickly within a few days after alleviating the stress, social defeat produces essentially permanent behavioral abnormalities. And that's particularly true for the social avoidance and metabolic impairments. Moreover, these long-lasting symptoms can be reversed by chronic non-acute administration of standard antidepressants. Anxiolytic drugs like benzodiazepines are without effect, and ketamine works acutely in about the same fraction of mice as the standard antidepressants. So we think that social defeat does offer some unique advantages in terms of studying stress responses in rodents. The point of resilience is particularly important because one of the challenges in the field is to understand when you subject a normal rodent to chronic stress, many of the adaptations that occur in the brain and body presumably are adaptive, are helping the rodent address the stress. And having the behavioral phenotype of resilience helps us parse what's adaptive versus maladaptive. And because of the longevity of these symptoms, because we can induce essentially a permanent behavioral syndrome in the mice after just 10 days of stress, our hypothesis has been that changes in gene expression and chromatin mechanisms are important, are one plausible candidate basis for these long-lasting adaptations. This concept is cartooned in this slide where we show the DNA double helix. For those of you who are not molecular biologists, we know that a mammalian organism has about 3 billion nucleotides in their genome. If stretched out linearly, that would be about 2 meters long. And somehow that 2 meters of DNA is compacted into a microscopic cell nucleus, an extraordinary achievement in evolution. And we've learned a lot about that organization, where the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome, which are then further compacted in the most compacted state into what we would recognize as a chromosome. And this is important because the activity of genes or other spans of DNA are dependent on how tightly that DNA is packed within chromatin. So at this level of analysis, DNA would be relatively active. In this level of analysis, the DNA is just too compacted to be contributing any biological information to the cell. Focusing at this level, now the DNA are these gold stripes wrapped around these histone spheres. Each one of these is a nucleosome showing the two opposite ends of the spectrum of chromatin structure: active chromatin where genes are active, condensed chromatin where genes are inactive. And we've learned a lot about the basic biology, which I'm just going to summarize quickly. We know that in active chromatin, spans of DNA are relatively exposed, which enables transcription factors, these are just some that we've studied, to bind to these relatively exposed regions of DNA. The binding of transcription factors to their response elements within their DNA regulatory regions of certain genes will recruit to those genes many other proteins, including histone acetyltransferases, which are the acetylate histones, and so on, to mediate the further opening of nucleosomes and the activation of genes. And studies in yeast and stem cells have provided sobering information about the complexity of these processes, with perhaps 200 proteins recruited to a single gene promoter in response to its activation, in response to an extracellular signal. At the opposite end of the spectrum, mechanisms of gene repression are equally complicated and are mediated by what are called repressor proteins, for example, HDACs, histone deacetylases, that would remove these acetyl groups, many types of histone methyltransferases, DNA methyltransferases, and so on, all serving to condense the chromatin. Starting about 10 years ago then, we decided that we did not know as a field enough about depression to make perfect guesses of what genes or proteins would be most important to study in our animal models. And we decided, with the advent of next generation sequencing technology, to take the most open-ended view possible. That approach is shown in this slide. We would use RNA sequencing to characterize RNAs that are up or down regulated in response to some experimental condition, say chronic stress. If those RNAs are induced through a transcriptional mechanism, they would be associated with some change in chromatin structure that's associated with the relaxation of nucleosomes and gene activation. We would study these chromatin modifications using ChIP-seq, chromatin immunoprecipitation, followed by deep sequencing, or a reduction in some repressive mechanism. And we would overlay on top of these RNA-seq data and these ChIP-seq data, ChIP-seq analyses for various transcription factors, which we know through other studies mediate susceptibility or resilience in particular brain areas. So this has been an enormous undertaking. The data depicted on this slide, just one time point, one brain region, all of these endpoints would involve something like 10 terabytes of sequencing data. So to be honest with you, we are still very much in the early stages of learning how to optimally mine such vast data sets, but we think that this is the right approach because unlike reliance on a single platform, which we know is associated with high rates of false positive and false negative discovery, when we focus in on overlaps of these different platforms in these Venn diagrams, we obtain lists of genes in which we can have much greater confidence for their regulation. And the vast amount of chromatin data provides a template for the biological mechanisms that are mediating changes in those genes' expression. So today what I'm going to do is give you a progress report of some of those data. The first experiment I'll focus on was done by Rose Baggett, and I want to thank Emmanuel Pierre-Thaman and Li Shen in our group for their bioinformatic expertise. What Rose did was to carry out RNA-seq in this experiment 2 days after 10 days of chronic social defeat stress. What I'm showing you are sequencing data for 4 brain areas: nucleus accumbens, medial prefrontal cortex, basolateral amygdala, and ventral hippocampus. Yellow are the genes that are induced, upregulated in resilient versus control animals. Blue are the genes that are downregulated in resilient versus control animals. And the bottom heat map simply shows how the same genes are regulated in susceptible versus control animals. The Venn diagrams show the same data, but just combine up or down regulation in resilient versus susceptible mice. So this kind of overview of this vast data set already can begin to help us formulate hypotheses of how to understand the biological basis of resilience and susceptibility. So for example, notice we had originally hypothesized that resilience was likely going to be mediated by the absence of bad changes that occur in susceptible mice. And although that's true to some extent, by far the predominant pattern is that resilience is the more plastic state, as shown in this slide. Many more genes are regulated in most regions, in particular this reward region, the nucleus accumbens, in the resilient state versus the susceptible state, so that susceptibility actually could be seen as a failure of adaptive plasticity. Another interesting high altitude pattern is that notice how in the resilient nucleus accumbens there's a predominant of gene induction. Very few genes are suppressed, whereas in the medial prefrontal cortex it's the exact opposite pattern. Many fewer genes are induced versus repressed, and we have data that reflects the underlying chromatin changes that predominate in those brain regions. Rose actually carried out that experiment at 3 different time points. I already showed you the data 2 days after social defeat. She has a data set looking a month after social defeat and a month after defeat in which the animals were exposed to an aggressor acutely. And what we asked the computer to do was to identify the predominant temporal pattern of gene regulation under each condition. That pattern is shown by these red lines, and the numbers in the graphs depict the number of genes that display those significant patterns. And again, this helps us focus where to look depending on what phenomenon we'd like to study. For example, if we wanted to understand what induced resilience initially at the early time point, we would be much more interested in the nucleus accumbens and the medial prefrontal cortex, where changes in gene regulation predominate at that early time point, largely go away at later time points. Whereas if we wanted to know what sustained resilience during that period of a month in between, after the defeat, we would be much more inclined to focus on the basolateral amygdala and the ventral hippocampus, where changes predominate at that later time point. Okay, so we think that it is absolutely essential, for the reasons I already described, to validate any work we do in laboratory animals in human postmortem brain tissue. So in parallel with the studies that I just mentioned to you, Benoit Labonté in my lab, who's now at the University of Laval, carried out a very large RNA sequencing study of depressed humans. This was a cohort of about 100 humans, half depressed, half not depressed, half men, half women, with a heterogeneous syndrome like depression. 20 an N of 25 in each group is not so big. On the other hand, this increases by about a factor of 4 the largest RNA-seq data set done on human depression previously. So this is just a start. We need to study many hundreds of individuals like this to get a complete understanding. We looked at 6 brain areas, several regions of prefrontal cortex, insula, nucleus accumbens, and ventral hippocampus. And one of the predominant findings that we made in this human data set were dramatic sex differences. There was less than 10% overlap in the genes that were abnormally expressed in all brain regions in depressed men versus depressed women. We have since validated the same phenomenon in our chronically stressed mice, largely different changes in gene expression across brain areas in males versus females. That's going to be the topic of my talk this afternoon, another shameless advertisement. So please come back, and we'll talk about some of the molecular basis driving these sex differences. But for this audience, it's really crucial to make the point that if these data hold up and are replicated, it suggests that depression may be a fundamentally different syndrome in men versus women, and really argues for drug discovery efforts, treatment efforts, to focus on women and men separately, as we know most efforts in the field in animals and humans have focused so far on males. It also raises the idea that we could validate animal models not only by trying to imagine whether their behavioral abnormalities are similar to human symptoms, but also using transcriptomics. That's depicted heuristically on this slide. This is not meant to be quantitative. It's work underway. I've mentioned to you so far our use of chronic social defeat stress. We've used 2 other chronic stress models, simply housing animals in isolation alone for a prolonged period of time, and then chronic variable stress, subjecting mice to different forms of stress every day for several weeks. And what we have found as we do RNA-seq analyses across brain regions, here are just 2 as examples, is that each of these chronic stress models produces largely distinct changes in gene expression across brain regions. There are some commonly regulated genes, but most of the regulated genes are different. Moreover, if we overlay the RNA-seq data that show gene expression differences in the different chronic stress models in mice with our human data sets, each model is roughly comparable to one another in recapitulating a fraction of the gene expression abnormalities seen in depressed humans, suggesting that no one animal model is perfect or any better than others, and what we really need in the field perhaps is an analysis of several animal models, each of which would, in a sense, capture a small subset of the human depression syndrome. And I think that makes sense given the heterogeneity of the syndrome and its complexity. All of the data that I've talked about so far is correlative in nature, and it's essential to establish causality. And I'm going to illustrate that with one particular gene that is beta-catenin, although we've done such validation for many genes. So let me just first introduce the beta-catenin pathway to you. Beta-catenin is a transcription factor. It is downstream of what is called Wnt signaling. These genes were all discovered in Drosophila based on morphological abnormalities upon their knockout in the flies. So they're given funny names. Wnts are secreted proteins. They stand for wingless in Drosophila. Wnt-secreted proteins act on frizzled receptors, which then act on a series of proteins called disheveled, which then eventually signals to beta-catenin. And when we drill into our data sets, this pathway is one of the most highly regulated in common in males and females, in mice and in humans, which is one of the reasons why we focused in on it initially. What our data suggested, just looking at our RNA-seq and ChIP-seq data, is that the pathway is profoundly downregulated in susceptible mice. Actually shows the opposite regulation. It's induced in resilient mice. This is all in the nucleus accumbens, that brain reward region. And it's also suppressed in depressed humans in this brain region. The different colors in the diagram simply depict different chromatin mechanisms inferred from our ChIP-seq assays. So Caroline Diaz, an MD PhD student in the lab, proceeded to study whether beta-catenin indeed is causally related to susceptibility versus resilience in nucleus accumbens. First experiment she did was to design a viral vector that overexpresses beta-catenin in the nucleus accumbens, and she found that it promotes resilience. That's shown over to the left. Animals that are subjected to 10 days of chronic social defeat stress show the reduction in social interaction. This is in social interaction with an empty cage. This is social or interaction with an empty cage. This is interaction when another mouse is present. So this is social interaction. You can see there's a reduction in social interaction or social avoidance in animals subjected to 10 days of chronic social defeat stress, injected with a virus making a control protein, green fluorescent protein. But that overexpression of beta-catenin produces a pro-resilient or antidepressant-like effect, reversing that phenotype. Caroline is able to show the opposite response. So now in response to just 2 days of social defeat, which by itself does not induce social avoidance in a normal mouse. So for example, a mouse normally will spend more time with another mouse than with an empty cage, not influenced by this 2-day social defeat paradigm. But already when we overexpress a dominant negative version of beta-catenin, a dominant negative is simply an antagonist of endogenous beta-catenin, we already begin to see a pro-susceptible phenotype, consistent with the fact that susceptible mice show a reduction in beta-catenin activity and resilient mice show an increase. Caroline complemented that viral study with a genetic approach. In this case, she took mice that had a floxed beta-catenin gene. A flox gene is a gene that can be knocked out in the presence of a bacterial enzyme called Cre recombinase, which we can express with a virus. So for example, normal mice, this is in response to submaximal social defeat, show their expected increase in social interaction with a mouse than with an empty cage. Where we knock out endogenous beta-catenin by virally expressing Cre, we see a pro-susceptible phenotype. And we can rescue that pro-susceptible phenotype by simply restoring beta-catenin levels with another virus, which the dominant negative beta-catenin cannot support. We can also analyze this on a cell type specific basis. So we know that nucleus accumbens predominantly contains 2 types of nerve cells, the D1 and D2 type medium spiny neurons, depending on the dopamine receptor subtype which they predominantly express. Looking at patterns of gene expression separately in the D2 and D1 neuron populations, we were only able to recapitulate the suppression of beta-catenin signaling in the nucleus in these D2 neurons in susceptible mice and resilient mice, in effect not seen in the D1 nerve cells. So this regulation seems to be specific to the D2 subtype of medium spiny neurons, which we can test behaviorally by using lines of D1 Cre and D2 Cre mice, where the virus only expresses beta-catenin in the Cre positive cells. So now using a virus such as this, overexpressing beta-catenin selectively in D1 neurons or D2 neurons, in mice all subjected to social defeat stress showing reductions in social interaction scores, only overexpression of beta-catenin in the D2 neurons restores the behavioral deficit. Overexpression in the D1 cells does not. In order to gain some insight into the genes to which beta-catenin is binding to mediate susceptibility and resilience, Caroline carried out ChIP-seq for beta-catenin in nucleus accumbens. The pie charts simply show the distribution of significant beta-catenin binding sites across the genome. You can see there's appreciable binding to promoter regions of genes, gene bodies, and about half the binding is outside of genes. It's an interesting finding from ChIP-seq data that all transcription factor bind about half to non-genic regions, and we're very interested in understanding what the functional consequences of that binding might be. But at this high level of analysis, one can already see that in susceptible nucleus accumbens, there's a reduction in beta-catenin binding sites, particularly at promoter regions, and an expansion in resilient mice. Looking at the same data with another tool, this is now looking at heat maps of beta-catenin binding. The center of each heat map shows the transcription start site of genes genome-wide, and then looking 5,000 bases up or downstream of those promoter regions. Each horizontal line on these heat maps would represent a different gene. So you can see the enrichment, the slight enrichment of beta-catenin at gene promoters, at the transcription start sites, but the appreciable induction of binding at promoters seen in resilient mice. Now, one of the interesting findings we had is that when we drilled into the specific genes that show these changes in beta-catenin binding, they were very different from beta-catenin target genes identified in other cell types. So other groups have done ChIP-seq for beta-catenin, mostly in cultured cells and in a few cases in peripheral tissues. And the binding sites are different. So the question is, why is it that when total cellular activity of beta-catenin is increased in a nerve cell or a peripheral cell, does that beta-catenin find its way to cell type specific targets? So the hypothesis is that what guides transcription factors to their particular sites in a cell specific manner is the chromatin landscape that pre-exists in that cell type for those genes. And some evidence for that hypothesis is shown in the other two heat maps. Here we're looking at ChIP-seq data for two types of chromatin modifications. This is a form of histone methylation. This is a form of histone acetylation. And you can see that the pre-existing landscape, both of these modifications are associated with gene activation, and these binding sites correspond quite well with the genes that show induction of beta-catenin binding in the nucleus accumbens of resilient animals, in support of our idea that these chromatin modifications are extremely important in providing docking sites for transcription factors. I don't expect you to be able to read the genes on this slide. These are an example of the target genes for beta-catenin. And what we're doing now is pursuing these target genes for possible insight into treatment opportunities. Beta-catenin itself, or the Wnt signaling pathway, cannot be used to treat depression. An activator of Wnt signaling or beta-catenin would likely cause cancer in many of our organs. So we need to identify those nucleus accumbens or brain-specific gene targets for beta-catenin through which beta-catenin mediates resilience if we want to advance this for therapeutics. And we have several projects in the lab pursuing that approach. Okay. Up until now, I have focused on the analysis of these ChIP-seq and RNA-seq data sets solely based on differential regulation. So in other words, with RNA-seq, looking at genes that show different levels of expression in one condition or another. I'm just going to take my jacket off. And because of the size of our data sets, we have the capacity to use much more complicated bioinformatic analyses. And for this, we've really benefited from having at Mount Sinai a great bioinformatic community led by Eric Schad, and this work was done in collaboration with Bin Zhang. And an example of that analysis is shown in this slide called weighted gene co-expression network analysis. Rather than focusing only on genes that show altered expression levels, we're actually looking at more complex relationships among genes in terms of how they show co-regulation. So it's organizing the genome based solely on data in our data sets on which genes are regulated together and perhaps regulated differently together in susceptible, resilient, or control conditions. So one way to show that analysis is on the right. This is called a Surcos plot. Each slice of the pie is called a gene module. It contains genes that our bioinformatics tell us are regulated together based on nothing known about the biology of these genes, just based on the temporal and spatial coincident regulation seen in our large data sets. Each gene could only be in one module. The most significant genes are shown at 12 o'clock, and these modules become less significant as one goes counterclockwise. One can drill into the genes contained within each module, identify leading gene ontology terms. For example, here are some interesting gene ontology terms associated with genes in the most highly ranked modules. We can also ask whether the modules contain genes that are also differentially expressed. So these inner concentric circles, the colors, depict significant regulation of genes in the 4 brain regions for which we have data in our mouse model. So I'm going to show you an example of how we've used these data sets. This is one of the modules, highly implicated in depression, very uniquely regulated in susceptible mice, not so much in control or resilient mice. So it's a susceptibility enriched module. Remember, the lines between the genes are just based on our data sets, not based on any biology. I'll point you to the key of the diagram. The solid blue circles are hub genes. Hub genes are those that are just connected with more genes, coincidentally. Red are the hub genes, but only serve hub functions, are only connected with genes in the susceptible state. Makes them more interesting. And the concentric light blue circle depicts whether those genes are also differentially expressed. So we focused on one particular gene that was highly regulated in this circuit. It is one of the strongest depression-specific hub genes, Sidekick1. We also chose it because it is suggestive as a possible depression risk gene in GWAS studies, although far from being significant genome-wide. And we also picked it because it was not differentially regulated. I wanted Rose, this is all work by Rose Baggett, I wanted Rose to study a gene that was implicated by this weighted gene co-expression network analysis, not by our differential gene analysis as a way to validate this bioinformatic approach. We also happen to have the tools available for Sidekick1 because my lab had previously demonstrated its involvement in cocaine responses in nucleus accumbens neurons. This network is largely in hippocampus, however. So Rose did the following studies to pursue Sidekick1, and I give her a lot of credit for following the bioinformatics, being agnostic and just following where the bioinformatics leads. The first thing she did was to test the effect of overexpressing Sidekick1 in the ventral hippocampus. What she found is that this induction of Sidekick1 made mice more susceptible to chronic social defeat stress. That's shown here, an increase in social avoidance to subthreshold social defeat upon overexpression of Sidekick1, and depression-like effects in other more standard animal assays. In collaboration with Yan Dong at the University of Pittsburgh, we studied what effect overexpression of Sidekick1 has on the activity of ventral hippocampal pyramidal neurons, and what we found was a robust induction of cellular activity as indicated by an increase in the frequency, no change in amplitude, of spontaneous excitatory postsynaptic currents. These data together would suggest that an increase in activity of ventral hippocampal pyramidal neurons would induce stress susceptibility. So that's a hypothesis that we could test through standard optogenetics. So what Rose did was to inject a virus expressing channel rhodopsin into ventral hippocampus. She was then interested in the influence of controlling the hippocampal projection to the nucleus accumbens, because we think nucleus accumbens is a crucial readout of the ways in which many cortical pathways control anhedonia, social behavior, and related behavioral responses. And the first experiment she did was to suppress the activity of that pathway by using low-frequency light stimulation in nucleus accumbens. So the virus is expressed in ventral hippocampus. we produce low-frequency light stimulation of nucleus accumbens, suppressing only those glutamate synapses and nucleus accumbens that come from ventral hippocampus. It induces, in a sense, a long-term depression at that particular glutamatergic projection. And what she found is that that was resilient. That was sufficient to make these animals more resilient. When animals are subjected to chronic social defeat stress, normal animals show the expected reduction in social behavior. LTD-like effects at that hippocampal projection to nucleus accumbens make the animals resilient. Rose could do the converse, now using 4 Hertz frequency of stimulation. This is a major frequency of stimulation exhibited by these ventral hippocampal pyramidal neurons in vivo. Now, in response to subthreshold defeat, which has no effect on social behavior, animals spend more time with another mouse than with an empty cage. Animals where this pathway is activated are showing a dramatic pro-susceptible effect, consistent with the deduction from our studies of Sidekick-1. More recently, we've been interested in pushing this experimental approach still further. How is it that increased excitability of ventral hippocampal neurons are creating a state of susceptibility? What are the circuit changes in the brain that occur as a result of that modification? We've been very fortunate to collaborate with Kafui Jirasa at Duke University, and these are data presented by his postdoc, Rainbow Holtman. And what these guys do is to record from multiple brain regions of an awake behaving mouse using standard tetrode recordings. I'm showing you data that they generated from 7 brain areas, many of which were included in the data sets that I've already shown you. And the blue lines are simply showing coincidence, coordinated circuit activity at specific frequencies in each brain region as seen with other frequencies in other brain regions. So in many ways, this is very analogous to the open-ended approach that we've taken with respect to gene expression studies. They're taking with respect to looking at coordinated co-regulation of nerve cells throughout the brain in these brain circuits. So they identified several networks, not surprisingly, labeled them randomly 1 through 6 or so. And one network in particular was quite interesting because it seemed to predict susceptibility in mice that had never before been exposed to social defeat. So that's shown over to the right. So what they did, this is before any exposure to stress, they measured neural activity in an animal's home cage, in the social interaction arena when there's no mouse present, an empty cage, and where there's a social target. And the strength of this network was significantly increased in those mice that were destined to become susceptible after 10 days of chronic social defeat stress. This, by the way, and I'll get to this in a moment, is one of the first abilities we've had to predict whether an inbred, genetically inbred C57 mouse is going to be resilient or susceptible. And this increased strength seems to persist after social defeat stress, but what is most dramatic is this this predictive ability of a stress-susceptible network. So what CAF and Rainbow then did was to adapt their tetrode recordings in a manner that would enable us to overexpress Sidekick-1 during tetrode recordings. That's shown in the upper left, where virus can be injected into the ventral hippocampus through these tetrode recording devices. Network activity was then recorded in the absence of stress, simply when the animals were in their home cage or in the social interaction arena with an empty cage or a social target. And what we found is that network 3, that network that I had shown you, was specifically enriched in animals that overexpress Sidekick-1 in the ventral hippocampus. That's shown here. Each line is a different mouse. Those mice injected overexpressing Sidekick-1 in ventral hippocampus, showing a significant induction of this pro-susceptible network pattern of activity across brain regions. So now the next step, beyond validating these data with additional animals, is to try to understand through which kind of connectivity do ventral hippocampal efferents coordinate this pattern. And then, of course, one of the major challenges in neuroscience, how does a pattern of neural activity really control behavior? But I do think that this story illustrates the approach that we've taken, and it's given me encouragement that starting out with very unbiased data can give us an indication of new mechanisms involved in stress responses. After all, this is all based on a gene that is not affected by chronic stress, right? So it was just implicated in our more complex data sets. In fact, when we published the earlier data, one of the reviewers asked us to ask whether any gene might do this. So we actually had to go back and reproduce all these studies with genes that were in the least significantly implicated module. Very painful, but it was actually a good control experiment. Those genes did not have an effect in these various assays. Okay, in my remaining 10 or so minutes, what I'd like to do is to shift gears and discuss the meaning of the word epigenetics and tell you about some other data that I think you'd find interesting. Up until this point, I've been using the word epigenetic in the most generic manner possible, simply referring to any change in chromatin modification associated with a relatively long-lived change in gene expression. But it was in the context of discussing epigenetics and what it means that we came up with other questions. One is, why do genetically inbred C57 mice show such divergent behavioral responses to the same social defeat stress? This is something we've spent a lot of time looking at. We've quantified the ferocity of the social interaction, the aggressiveness of the aggressive mice. We cannot see any distinction in the behavioral experience, as best as human eyes can tell, between the animal that becomes susceptible and becomes resilient. We know that genetics is important because if we study different inbred mouse strains, each strain has its own inherent rate of susceptibility and resilience that's very different across strains. But in our case, genetics is probably not the explanation because C57 mice are 99.9% genetically identical. We also know that environmental factors are important. In fact, this afternoon, I'm also going to be talking about what we know about how we can dial up or down susceptibility based on the behavioral experience of the mouse. But in the standard experiments we do, the mice are treated in as identical a manner as possible. I mentioned the social defeat experience is non-detectable, is not detectably different. There is no relationship between dominance hierarchy of the mice and susceptibility and resilience. So we thought perhaps dominant mice or submissive mice may be more or less susceptible. That is not the case. We've quantified maternal behavior to these animals. We have not been able to detect anything significant. Each mother will give birth to and raise roughly equivalent ratios of susceptible and resilient mice. And we have not found any measure, except the electrophysiological measure that I mentioned previously, which is pretty complicated, any way to predict which mouse will become susceptible or resilient based on blood measures, a battery of behavioral tests, and so on. So that led us to another meaning of the word epigenetic, and which is closer to the use that Waddington had when he first introduced the term 60, 70 years ago, which is that during development, that as 100 billion nerve cells are formed in a human brain and 100 trillion synapses are formed in a human brain, a lot of random stuff happens. For example, patterns of cortical gyruses are very different among identical twins, thought to reflect differences in development that occur stochastically through random mechanisms. Actually, that might be a major reason why it's been so difficult to solidify the genetic basis of psychiatric syndromes, because much of what we may be seeing as psychiatrists could relate to these random changes during development, which are very hard to study. In fact, we're very interested in looking at this experimentally in our mice, but we would need a way to predict which mice are going to be resilient or susceptible with pretty good assurance. And that's why we're also very interested in the electrophysiological data, because they may provide us an ability to know, okay, here's a mouse, adult mouse. Is it destined to be susceptible or resilient? Now let's characterize its chromatin epigenetic state compared to a genetically inbred mouse that's going to be resilient, and then compare the two. So that's something that we're interested in doing. But it was in these conversations that people in the lab also came up with a third meaning of the word epigenetic, and that is the transgenerational transmission of behavioral experience to offspring. That's something that shouldn't happen. Jean-Baptiste Lamarck was, what's the right word? Disrespected or proven wrong, whatever, 150 years ago. But there's been a lot of data, as you know, over the last 5 or so years showing that if you do something to a rodent, you will see differences in the offspring. So guys in my lab, David Dept and Quincy LaPlante, did an experiment that I didn't even know they did. They took male mice, they bred them, they subjected them to control conditions or 10 days of chronic social defeat stress, waited a month, bred the males with normal females. As soon as the females were pregnant, they took the males out of the cage. Females raised the babies, both males and females, to term. We studied the mice as adults, and lo and behold, there was a dramatic pro-susceptible phenotype in the offspring animals, right? Not supposed to happen. I thought this was crazy. Made the guys repeat this. Once I found out about it, made them repeat it again and again. But what was clear, and it's a robust, one of the most robust phenotypes we've seen, is that the sons of control mice show normal responses. They show more social interaction with a mouse than with an empty cage. But there was a dramatic pro-susceptible phenotype in the sons of defeated fathers. So at this stage, we figured, okay, we're going to nail down an epigenetic mechanism by taking the sperm of these defeated fathers, putting the sperm in vitro with eggs from normal females, get embryos and plant them in other normal females, and then we'll compare the IVF offspring of normal fathers and defeated fathers. So this is when, as a PI, the people in your lab say, you know, thanks a lot, right? And there were phenotypes in these IVF offspring of defeated fathers, but they were extremely subtle. No phenotype in social defeat. There were modest, very modest phenotypes in other behavioral tests. So there was some evidence, but not nearly robust enough to study. So this was published in 2011, and upon reflection, we thought of two caveats, which we only realized in retrospect. The first is a behavioral caveat, that we know that there's an incredible literature in rodents and and actually non-mammalian vertebrates also, that mothers will impart different amounts of maternal behavior and resources to offspring based on the male with which they procreated. So maybe, and my wife hates it when I say it like this, and particularly in Trump's America, maybe the mothers knew they had sex with losers, right? And I'm, not a behavioral psychologist, but it was something worth considering. But we also realized that we did not do justice to the possible epigenetic mechanism as well. Because again, in retrospect, by taking sperm and egg and putting them in vitro, we know that we're allowing a lot of epigenetic reprogramming to occur, so it was not the best experiment. So more recently, Dina Walker, another postdoc in the lab, did another experiment, which I never thought we'd do in my lab. So this is what Dina did. She took normal males that were vasectomized, had them have sex with normal females. Right after the sexual intercourse, Dina artificially inseminated the females with sperm from either the defeated or control mice. And lo and behold, she was able to recapitulate the phenotype. So this is an example shown in social defeat. Similar phenotypes in other behavioral testing. Now, no question, the magnitude of the effect is much larger with normal sexual reproduction. So we actually do believe that there is a behavioral effect here of the mice, mother mice changing their care of the offspring in some way based on the sexual experience. Nevertheless, now that we have the vehicle, now that we've demonstrated that sperm from defeated fathers can be an active vehicle of inducing a behavioral susceptibility in offspring, male and female mice, we can now study the mechanism occurring in those sperm cells. Based on work by Tracy Bale at Penn and Isabel Mansui in Zurich, both of whom have provided some evidence that chronic stress alters microRNAs, either somehow adhered to the outside of sperm cells or perhaps even inside of the very small sperm cell, might be transmitting some stress susceptibility to the next offspring. So we're now in the process of doing microRNA sequencing, as well as considering other epigenetic mechanisms. I think we have to be skeptical as a field in believing this until we really nail down a mechanism. Because again, molecular geneticists are extremely skeptical because according to what's known about meiosis and epigenetic reprogramming, this kind of thing should not occur. Yet I think this is one of the best examples where at least we know something about the sperm cells can be that active vehicle. So with that, let me conclude. I hope I've convinced you today that by using a broad range of big data approaches across several animal models, overlaid with analysis of human postmortem brain tissue, it is possible to gain new insight into the neurobiological mechanisms that contribute to a syndrome as complicated as depression. This morning I focused on mechanisms of stress susceptibility and resilience. This afternoon I'm going to be focusing on examples where these changes mediate dramatic sex differences in depressed men versus depressed women, and also how behavioral experience controls susceptibility and resilience for a lifetime. We do believe that chromatin mechanisms are a very appealing, attractive manner in which long-lasting behavioral change can be mediated. I've shown you some examples of that. And I'll end with a reflection on a 30-year anniversary of having my lab, which is a very humbling one. And that is that over the past 30 years, even though the field, and I'd like to think I contributed something to the field, has learned a lot about the brain and maybe a lot about stress responses and maybe a bit about depression, we have no better treatments. We have not substantially improved our ability to care for people with depression or other psychiatric syndromes in a dramatic way. So that's sobering after looking back on one's career for 30 years. And I just hope, and this is for the younger people in the audience where the torch now gets passed, is that I'm hoping that the lack of progress simply reflected the enormous and unpredicted complexity of the brain and the genetic underpinnings of biological regulation, which are far more complicated than we imagined 3 decades ago. And the hope is that as the tools of neuroscience and molecular biology improve, we will be able to identify at long last ways to better treat our patients. I'd like to thank our funding sources, NIMH and Hope for Depression Research Foundation. And I'll just leave you with a slide from my hometown. This was taken by a friend of mine from an airplane. This is Central Park. This is Mount Sinai on the Upper East Side. And I like this slide because this is the Metropolitan Museum of Art. And I don't know if you can see this orangish building right there. That's my apartment. Anyway, thank you all very much.
Yeah, thank you so much for a wonderful lecture and also a voyage through a very, very complex field that is not easy to dumb down for some of us, right? So thank you very much. I want to check, though, is the message I should get out of it as a defeated psychiatrist who's unable to figure out depression, that dot, dot, dot.
One of the funniest responses I got.
joking.
No, I know, but I'll tell you a vignette. I gave grand rounds to a surgery department. By the way, 8 a.m. is painful. I'm glad I'm on East Coast time. I had to give surgical grand rounds at 7 a.m. on the East Coast, which was really painful. That's why I went into psychiatry, so I wouldn't have to do that. The chair of surgery asked whether social defeat was a model of surgical residency.
It used to be. I mean, now there are more resident rights. But anyway, I'm sure there are lots of comments and questions. Is that okay?
Sure, of
We can
course,
go
yeah.
around a
Happy to
bit,
answer any
and
questions
please
you guys
wait
have.
for the microphone.
Okay.
Thank you for this excellent presentation. For those of us who won't be able to hear your
Yeah.
sex difference talk, I'd love to hear the highlights.
No.
And then, and then I'd be interested in your thoughts on the work coming out of Lance Kriegfield's lab in Berkeley, looking at, he takes female pregnant mice, and if he disrupts their circadian rhythms, this is not a stress effect. There, in the offspring, there is impaired growth, impaired social behavior, and increased anxiety-like behavior. And so that whole...
Sure, I'll just give you a quick headliner for the afternoon's talk. I mean, basically what we've done is to drill in to the gene differences that are apparent between depressed men and depressed women, or stressed mice and males and females, and identifying specific genes that illustrate those differences, and then actually identifying tangible hypotheses through which we could induce selective antidepressant effects in women, effects that would not be seen in men, and vice versa. So we think that we actually have empirical data supporting, based on human and animal investigation, that sex-specific treatments for depression may be appropriate. I'm not familiar with the person's studies that you mentioned. Do you know whether, so these were pregnant females that were, yeah. So the challenge in epigenetic studies, and the reason why most laboratories study paternal transmission as we did, is because epigenetics is one generation easier to study in males versus females. Because when you study pregnant females, the test mouse is present, is exposed to everything that the mother was exposed to. So that's not necessarily epigenetic. That could be a physical effect of the fetus in the womb that then is displayed for the rest of the animal's life. May be mediated through epigenetic mechanisms, stable changes in gene expression as the animal develops, brains develop, but it's not it may not be transgenerational transmission of a behavioral experience. I don't know if that makes sense. So what you described is an example of, is one of many examples where pregnant females, challenges to pregnant females produce dramatic changes in their offspring. Again, whether that's through epigenetic transmission or direct effects on the fetuses remains unknown. Very few have taken that to subsequent generations. There are some examples, for example, in the feeding literature with starvation or overeating, that mothers exposed to those wide ranges of diet do show changes in their offspring's metabolic state for several generations. So the implication is that that would be a true example of epigenetic inheritance, but the molecular basis of it is unknown.
Erica,
Yeah,
wonderful, wonderful
Thanks.
talk. Two quick questions. Strain differences in mice and rats and rodents and people are profound. You've already alluded to the fact that there's differences in resilience and susceptibility across different strains. How do you know you haven't just identified the biology of depression for C57s and elsewhere? The second part question is the circuitry that you're studying, near and dear to my heart, is one that's important in regulating all of the phenotypes that you're seeing in your defeated mice, having to do with activity, goal seeking, pleasure seeking, etc. And how do you know that the changes that you're seeing in the circuitry aren't an epiphenomena of the phenotype that's being generated?
Right, okay, yeah. So in terms of the C57 question, all the work that we do now in animals is based on C57 mice. And it's an interesting dichotomy in approach between traditional molecular biologists and traditional behavioral scientists, most of whom would prefer to study outbred animals or at least compare different inbred strains. Because you're absolutely right, focusing on a single inbred strain will tell you about the genetic background of that strain and might be completely irrelevant to the human condition. The way we address that is by not studying a gene from animal models from C57 mice unless we can validate it in human postmortem brain tissue. And we think that that provides at least some reassurance that the genes and mechanisms that we're studying are related to the human condition. But I think that the other approaches of focusing on outbred animals like Abe does or other approaches are absolutely essential to make sure that one does not fall victim to that trap. The question of whether a behavior or the circuit comes first is obviously a really tough one in neuroscience. I think that that is the perhaps the major challenge in the neurosciences today, as I mentioned. How do we don't even know, when I lecture the medical students, we don't even know how the visual perception of a tree, for example, we can kind of figure out how that's projected in our visual cortex. But how the association and then prefrontal cortex determines whether we like the tree because we're an environmentalist or we want to make money on the tree by cutting it down is completely opaque to a neurophysiologist. We have no knowledge of the circuitry of that, right? What the tools today allow, though, which is really exciting, is that perhaps with optogenetics, with various tools of capturing ensembles of nerve cells, like with calcium imaging or something like that, and then going back into those same animals and then manipulating just those ensembles of nerve cells optogenetically or chemogenetically, and then looking at a behavioral output, we might be able to get to the chicken or egg kind of question that you're asking. But I agree, it's a very important one.
Thank you for a wonderful talk. Could you say a little bit, there's a lot of people in the department interested in psychoneuroimmunology. So could you say a little bit about the role of inflammation and immunological factors and how they interact with any of the epigenetic findings that you gave us?
Yes, absolutely. So when we look at our data sets, very initially, one of the strongest families of genes that showed regulation in animal models and in human postmortem brain tissue are genes that are implicated in the immune system and inflammation. My lab has not further pursued those mechanisms. Scott Russo, a faculty member at Mount Sinai, has made that his focus and has beautiful data, which led him down a very interesting path of not only regulation of immune genes in the brain, but then how that correlates with regulation of the immune system in the periphery, and how a pro-inflammatory state is seen in a subset of susceptible mice, just like it's seen in a subset of depressed humans. And then by manipulating the peripheral immune system, he can actually produce an antidepressant response in those mice. And we're actually studying at Mount Sinai now an anti-interleukin-6 antibody introduced for the treatment of peripheral rheumatologic disease for depression, specifically in those patients, mostly women, who have evidence of a hyperexcitable release of IL-6 from their peripheral leukocytes in the depressed state. So it'd be a really interesting. This is with J&J, by the way, using their antibody. So I think it's an extremely timely and important focus of investigation. And from a biological point of view, I think one of the most interesting questions is, how does the peripheral immune system get regulated under these conditions? Why is bone marrow showing such clear responses that differentiate susceptible and resilient mice? Is that all through the sympathetic nervous system or circulating hormones? How is it that pro-inflammatory molecules in the blood get into the brain? Once they're in the brain, how do they influence nerve cells and non-nerve cells? How is communication between glial cells, endothelial cells, and nerve cells controlled by these cytokine mechanisms? Again, a really relatively understudied area in neuroscience in terms of which cell type secretes which hormone and acts on a receptor on another cell type. So it's a great area of investigation.
Hey, Art, go ahead.
Hey,
Yeah, that was really
Terrific.
Thanks.
I'm really intrigued by the susceptibility resistance thing, and I was really glad when you got to the end of your talk and you really addressed that head on, like how is it that these inbred strains could be so different? Do you think part of your ability to get susceptibility and resistance is just a dosage of the stress? Like if you gave them chronic mild stress, or I'm sorry, the social defeat stress for 30 days, would everybody be susceptible at that point?
So we, yeah, go ahead.
And also C57 or black 6, as you know, there's lots of substrains of that.
Yes.
And one thing that we're working on in my lab is we're resequencing all of those, and we're actually doing EQTL mapping in them, because even though they're incredibly similar genetically, they have what's more similar to de novo mutations in humans. They have very subtle accumulation of mutations over time, and yet those have big impacts on behavior, and I wouldn't be surprised to hear that they would change the ratio of susceptible to resistant, and then you'd have a tractable genetic system where there were really maybe 10,000, 100,000 mutations, most of which are completely neutral, and you might actually be able to disentangle.
Right, so in terms of the last question, I totally agree. We know that we need to get C57 mice from the same supplier. Not only the same supplier we use Jack's labs, but it has to be from the same facility of Jackson, because they will differ depending on where Jackson is breeding the mice, as you know. I don't know if this is something that Stephanie and you are already going to do, but if not, perhaps that's something we could do together, because it would be really interesting to overlay your genome sequence and EQTL data on some of the stress-induced changes. So we can all talk about that. Yeah. Yeah. What was your first question? Oh, yeah. Right. Right. So we've spent a lot of time looking at that. It's probably a bit of both. So we definitely see the ratio of resilience going down the more the animals are subjected to social defeat. There does seem to be a core number of mice that remain resilient despite being subjected to really an extraordinary amount of defeat, and we're sort of limited by how much defeat we can do by our animal care overseers, appropriately so. So that's future, right? Exactly. Yes, exactly. That's right. No defeat, resilience. So so we think that it is partly a dosage effect, because mice do succumb, and particularly if we repeat them. If we expose the animals to repeated bouts of defeat separated by a month or two, a subset of resilient animals peel off and become susceptible. But there is this core group of mice that really look like they're just inherently resilient. Stephanie? As much as a small litter can tell you. That is the one possibility that that is one possibility that we have not looked at, just because my lab can't do that. I mean, maybe that's something that you guys could do, because one can go into a uterus now, mark the animals for their positioning, and then study them. So that would be very worth doing. Really cool.
So it was a really nice talk, and I just have a question coming back to early part of your talk, where you showed the overlap or non-overlap between the various in gene expression from the RNA-seq data, I believe, between various mouse models of depression that were generated in the humans.
Yeah.
And you focused on, you mentioned how there's some similarities but lots of differences. But maybe the core thing to think about are the things that were identical in all of those. And have you actually looked carefully at those as potential genes that really are common to different forms of depression, including in humans? So could that give you a good angle on really something to pursue? And you didn't say anything about that.
Yeah, no, absolutely. So there were two ways to think about it. One way is just as you said, that if you look across mouse models and overlay rat models too, right? And find there will be some mechanisms that will be seen across these models and species. Validate that with human. Look at the things that are in common between males and females too, because we're talking about hundreds of genes. There are going to be genes that will survive those kinds of overlays. Maybe those are the most important. When we've done that, there's no question that those genes are really interesting. In fact, those are a lot of the genes that people have studied. Like BDNF is one of those most highly ranked genes. Beta-catenin, as I mentioned, is highly seen across models, across sexes, and others. Delta phos B is another example. CREB. But perhaps that is still the counterpoint is that perhaps that gives short shrift to the fact that two people with depression are very different from one another, and that our goals of lumping and focusing on what's common have not met with such great therapeutic success, although they've met with great neurobiologic success. Maybe we need to parse the complexity more than we've done. And I don't know if that's going to be more effective at developing better treatments, but I think it deserves a chance.
Hi, Eric. Thanks for a terrific talk. I'd like to follow up on a couple of the comments related to social defeat among surgical residents. Our own residents are very resilient, and I think you'll probably meet some after this talk. But based on the idea of 30 years of progress and yet the slow kind of pace for new therapeutics, what advice can you give to those who are starting their own path and their own careers dedicated towards finding the cures or even improving the care of our patients?
Thank you for that question, because I do tend to be too negative sometimes or too nihilistic sometimes, and it's really important not to be. When what really bothers me, and I hope I live to see the day when we can make changes for our patients, when we look at international or national data, the impact of mental illness on humanity is extraordinary. And as we do a better job of treating cardiovascular disease, cancers, infectious diseases worldwide, the impact of mental illness will just increase further. We've seen that in various World Health Organization assessments. The most recent data I've seen suggests that something like 40%, 40%, of all lost productivity in the United States is due to mental illness. Most of that depression and anxiety. So these are just extraordinary challenges for us. And what I'd say to the young people is, what I always say to the young people is, never listen to the whining and pessimism of old people. Because optimism is a crucial piece of resilience. And optimism is necessary in order to succeed. And the kinds of tools that the young people have available today are so much more penetrating, transformational really, compared to what we couldn't even have imagined 30 years ago. That the ability to study the brain in animals and humans and overlay different levels of analysis from molecules to circuits to behavior in animals and humans is unprecedented. And so I remain optimistic that we'll get there one day. I'm just frustrated for myself.
Hi, thanks for the great talk. I was just wondering what we know about the opposite effect, like of previous enrichment. Does if mice are previously having a highly positive experience, does that affect the ratio of how many become susceptible or resilient? Or if there's a period of enrichment between two bouts of social defeat, does it affect their performance in the second bout?
It does. Environmental enrichment, so housing mice with a lot of other mice with a lot of toys in the cage, will make the animals more resilient to social defeat and other stresses. But the effects don't last that long. So we haven't studied it because we were really interested in mechanisms of long-lasting change. And it's been very difficult for us to take mouse pups and do things to them, or allow them to do things to themselves, that institute a lifelong increase in resilience. We have not yet succeeded in doing that. However, we've done the opposite. So we have found that certain types of early life stress, we'll talk about this afternoon, only when timed at a particular period in development, will essentially produce a lifelong increase in susceptibility.
And with respect to the epigenetic work that you mentioned, have there been work with the females that have been defeated prior to fertilization and see if you can tease apart any effects of
so maternal transmission of social defeat?
Yes, but before the dams are pregnant.
Right. No, we have not done that. One important advance is that we and other groups have now established social defeat in females. It took a while to do that, but we've been able to get males that are aggressive enough to actually fight with females. So we validated female social defeat, but have not yet looked at maternal transmission to next generations.
I know you sort of alluded to this, and maybe you can shed more light on it, but I wanted to ask if there were any age differences in terms of if you tested, it seems like some age groups with this defeat model, and were certain ages more resilient than others, where maybe even you have these susceptible mice, but if you do it later in life, they're able to overcome.
Yeah. So Carlos Bolaños, who's a former postdoc of mine, now at Texas A&M, is looking at that question, which is very interesting. In order to look at it, it was necessary to develop a variant of social defeat that did not involve the actual fighting, because if the aggressor mouse is much bigger than a younger mouse, the experience of the defeat may be very different than for an adult test mouse, for example. So what Carlos and Brandon Warren, his graduate student, did was to develop what we call witness social defeat. So you take a C57 mouse, put it in a cage across a screen from an aggressor mouse defeating another C57 mouse just for a few minutes. That physically defeated mouse is taken out of the cage, and now the witness mouse is living with the aggressor for the rest of the day. That witness mouse develops an equivalent syndrome as the physically defeated animal. It's very, very similar. We've done RNA-seq, and there are a lot of similarities between the two conditions. So that's an ideal system to address your question. And the preliminary data he has, interestingly, is the opposite of what I'd expect, is that adolescent animals, that's being anthropomorphic, maybe 40, 50-day-old mice are more susceptible. I thought they'd be more resilient. They're more susceptible to witnessing defeat. So that's interesting, and may have some implications for stress susceptibility during adolescent development. So something we're very interested in looking at.
Eric.
All right, Ming,
Oh, no,
it's all up to you.
Thank you, Eric. We are in the council. Yeah, we are in the council together. So we share all kinds of same opinions. Okay, brief. Yesterday we met, and we are trained in medicine. We always try to find pathology rather than resilience or well-being. That is the future of our research. And I admire that you can do it from your preclinical for fertilization here and there. For the human is very difficult. So I'd like to ask you a question.
Sure.
Yesterday I talked to you about the PTSD. Before deployment, we already correct the RNA gene expression, and we found that even exposed to stress, they are immunological and inflammatory gene actually are dysregulated. Now we are going into study resilience on those who came back and no PTSD symptom at all. Do you have any suggestion? We cannot do the experiment which you have done, but from your work, can you recommend anything which we can do to study resilience and well-being?
Well, sure. So I think, one of the things you could do is that the troops who came back and saw equivalent combat to the men and women who did develop PTSD, but in this case did not, is to look at time-dependent changes in their peripheral gene expression. For all we know, there may be changes in peripheral immunity that are uniquely associated with resilience.
Yeah.
We're very interested in looking at ways to recapitulate mechanisms of natural resilience, as you said, in susceptible individuals, as well as many other metabolic insights that you might get from those studies.
Thank you.
The notion of looking at natural resilience is essential. That there are examples of people who harbor disease-causing mutations in amyloid precursor protein or presenilin, for example, which cause early onset Alzheimer's disease, yet there are individuals that harbor the same mutations that don't get sick. So it's not complete penetrance. And so those people must have resilience factors that protect them. So I do think that studying resilience offers enormous benefit.
It's supposed to be a brief question.
Yeah, yeah, no, it's. Mm-hmm. Mm-hmm.